All right, we still have some folks entering the room, but we'll go ahead and kick it off. Welcome to the final seminar of 2025 for the N of One Collaborative Seminar Series. Thank you so much for joining us. My name is Nicole Nolan. I'm N1C's Programs Manager. I'm just going to share a few brief housekeeping points, and then I will hand it over to Dr. Tsai, who is moderating today's session. Um, Importantly, we as N1C really want to thank our partners, the Chan Zuckerberg Initiative, HanGene, and ChemGenes, who provide mission critical support for N1C, including support of this program, the seminar series. If you or your organization would like to discuss how you can support N1C, please feel free to email me. I will drop my email in the chat and I would love to hear from you. Secondly, I would like to issue an invitation to everyone on the call to join us on Connections following the seminar. If you have any questions or thoughts after today's talk, Connections is our new virtual space where we collaborate virtually across the globe, across the year. So please join us. Without further ado, I'll hand it over to Dr. Tsai.
Terrific. Thanks, Nicole. I'm Shandar Tsai. I'm an associate member in the Department of Hematology at St. Jude Children's Research Hospital, where I work on genome editing technologies towards therapeutics. I'm also a co-chair of the steering committee of the NIH Somatic Cell Genome Editing Consortium, which is where I met PJ. Some kind of opening notes. We want this to be an interactive session, so please turn on your cameras if you're able to, especially during the moderated Q&A. If you have questions, please put them in the chat for discussion after the presentation. This will be recorded, so you can view it later on the N1C Connections. You will automatically receive a survey at the end of the seminar. It will pop up when you close the Zoom link, and so we'd appreciate your feedback to improve and host topics that everyone's interested in. And with that, I'd love to introduce PJ. So PJ is the acting director of the NCATS Division of Rare Disease Research Innovation. And so he represents NCATS in wide-ranging gene therapy working group. He's also the coordinator for the NIH Common Fund, including the Somatic Cell Genome Editing Consortium. He was selected as the recipient of the 2022 Sonia Scarlatos Public Service Award by the American Society of Gene and Cell Therapy for consistently and fostering and enhancing the field of gene and cell therapy. PJ is also the person where I learned about the meaning of zebras in rare disease research as well. So there's an old medical adage that, where you hear hoofbeats, think horses, not zebras. And I think zebras have become a symbol for rare disease patients because it's important not to forget them in the context of common disease. And so with that, I really look forward to your talk, PJ.
Great, thanks, Shangdar. I forgot I should have put one of my zebra ties on, but I wasn't thinking. So thanks for the opportunity to speak here. I'm looking forward to letting you know about some of the work we're doing supporting therapeutic platform strategies to treat monogenic disease at NIH, and also look forward to the questions and discussion. So here are my disclosure statements: no financial conflicts, and any opinions are mine and not those of the NIH or Health and Human Services. So what we think about very often all the time in the rare disease field is the rapidly increasing number of disorders with a known molecular basis. It's been estimated now that it's over 10,000 or so. Kind of depends on how you do the lumping or splitting, but it's clearly in the thousands, and it will be increasing as we do more whole genome sequencing. But the number of approved therapies for all of those diseases is lagging far behind, estimated on the order of 600 or so. And the original director of NCATS, Chris Austin, once calculated that at this rate, it would take 2,000 years to come up with approved therapies for all of these rare diseases, which was a good line, a memorable line, but it presupposed that anybody would actually do that, that people are actually trying to develop treatments for all of these rare diseases. And the reality is something different. This is from work from a European group published a few years ago, where they kind of broke down rare diseases into different levels of prevalence. In the United States, a rare disease is one that affects less than 200,000 people There are somewhat different definitions in different countries, but the basic idea still holds that of all those rare diseases, there's a fairly small number, about 150 or so, that have the highest prevalence, on the order of 1 in 10,000. And those not unreasonably get the majority of financial and commercial interest in terms of drug development. But the far, far greater percentage of those of rare diseases are those that are on the order of the 1 in a millions, what are sometimes referred to as ultra rare diseases. I don't particularly like that term, but you can think of it as it is. And these ones get very little commercial interest. And the most obvious example of the importance of this is spinal muscular atrophy, which affects about, let me see, I think it's 25,000 people or so in the US. And that has gotten a lot of interest, three approved approaches for that. And it's interesting to think about what would have happened if instead, I think it's about 50,000 patients. What if there were only 50 spinal muscular atrophy patients? They would still be dying at the age of 2 without any treatments. And that's the importance of prevalence. And this is not stated as a way of criticizing for-profit drug companies. I benefit from for-profit drug companies every morning when I take pills. You know, you get to my age, you take a lot of pills. They work really great. I'm not trying to criticize them. It's just a matter of sort of dealing with reality and thinking that if we are going to ever get to all these diseases, we really need, I think, a very different way of thinking about the problem. So one way to do that is to really kind of think about grouping diseases on the basis of the underlying etiologies. And indeed, even though there are thousands of rare diseases, when you really get down to the underlying etiologies, which is what we develop treatments for, the number is much, much smaller. And if you think about monogenic diseases, these are what I think of as sort of the big four of the mutation types that are actionable: nonsense, missense, abnormal RNA splicing, and dominant diseases. There's also sort of signalopathies that can be treated with drugs that impact those. So one approach is to try to focus on those and group patients along those lines. And we have different drug approaches to do that, as shown here. Probably one of the most striking examples of that, of an approach to treat what I at least think of as cryptic splice site mutation disease, is, of course, the work on mucin that Tim Yu did along with Julia Vitarello, and this really landmark study. I'm shocked now, it goes all the way back to 2016, time flies. They were able to go from a diagnosis of MULA in all the way to an actual first patient dosing in about 2 years. And this is done really without focus on the actual specific of the disease, but simply on the basis of the molecular genetic abnormality. And I think this approach is one that can be used and is being used by Tim and others to do something similar. And as an example of, I think, what makes sense as a much better way to approach these many, many different rare genetic diseases. But at even higher level of etiology, so to speak, for almost any monogenic disease, there are some obvious approaches which are applicable, and these are gene therapy and gene editing, and that's a lot of what I'm going to be talking about today. So in thinking about these approaches on therapeutic platforms, which we began to do years ago at NIH, one of the first things we did was to develop the Platform Vector Gene Therapies, or PAVE-GT program. The idea here was, could we utilize this, one AAV vector stereotype to treat multiple diseases? Originally, we were thinking to do AAV9 and to treat 2 organic acidemias, 2 metabolic diseases with Chuck Venditti at the NIH Clinical Center, and 2 neuromuscular junction disorders with Karsten Bonneman at the NIH Clinical Center. And the idea was, if we could kind of do this and keep that one platform vector, we might be able to speed the process of getting to IND filing. But also we wanted, in the process of doing this, we wanted to sort of learn from those regulatory interactions and make the information public to benefit the whole field. And we have been doing that. It turns out now we're actually going to use two different serotypes, AAV9 for the metabolic disease and AAV8 for the uh, neuromuscular junction disease. But the basic approach is similar. And indeed, we have been able to publish some of these regulatory interactions, first focusing on the orphan drug and pediatric designation for AAV9 for propionic acidemia, and then also discussing and making available the FDA interactions in terms of the pre-IND meeting. And this is a theme that I'll come back to time and time again throughout the course of this talk. So moving forward, a kind of similar approach was developed, and this is what's called the Bespoke Gene Therapy Consortium. This is an Accelerating Medicines Partnership, or an AMP, that is supported by and organized by the foundation of the NIH. And a key point here is that the foundation for the NIH is actually not part of the NIH. It's not part of the government. It exists independently to allow government researchers and government-funded science interact with private industry in ways that would actually not be legal otherwise. So it's a really wonderful program that was begun a few years ago. Again, organized by the NIH, and I'm one of the co-chairs along that with Tim Miller from Thermo Fisher, one of the companies involved. It is, as you can see here, a very broad public-private partnership involving about 10 or so different entities within the NIH, as well as for-profit and non-profit entities. As you can see on the right, roughly half and half in terms of the financial donations. I want to have a special shout out to the California Institute of Regenerative Medicine and CIRM as one of our partners who have really stepped up to actually support some of the projects, allowing us to do more with this effort. And there are roughly, or really, when we developed the BGTC, we tried to create it in terms of two sort of work streams. One was sort of more focused on some work in the basic biology of AAV, either how vectors are produced or potentially how we could enhance therapeutic gene expression once they go into patients, with the goal of enhancing efficiency on both of those. But the biggest part of the BGTC that really takes the most time and effort, and we hope impact, is to really focus on making the whole process of going from the idea and a proof of concept study in AAV gene therapy all the way to first in human clinical trials. And a lot of that is to sort of try to harmonize and streamline how we navigate that regulatory process. And a big part of this too is to try to identify some minimal packages of things like critical quality attributes for AAVs that might be produced. How do we know that they're safe enough to go into people? What are the minimum data sets that you need to determine that they're safe to go into people in first in human studies? You might have thought, as I thought, that that was already well specified in legislation, but it isn't. So we've been trying to explore that as part of the BGTC. And also along a similar lines, what can we find out about the minimal set of GLP toxicology studies that are necessary to show that an AAV is safe? And here again, it's really kind of working with the groups of experts from these different companies I showed you to kind of brainstorm and come up with these minimal packages and minimal approaches and bring them to the FDA and get their feedback from them. And ultimately also here again, make that information publicly available. So this is our current BGT project portfolio. We've got 7 different diseases that we're currently working on. Some of them are primarily funded by CIRM, and that's indicated there. One of them is currently sidelined, but we got these 7 moving forward. Quite intentionally, we have 3. We have different tissue targets because we want to explore some of the challenges for different tissue targets. So we have 3 ocular diseases, 3 neurologic disease, and 2 where we're going to be going systemic delivery. Very recently, we got some really good news with regard to CMT4J that the CIRM has been able to provide the funding for opening the clinical trial, which we anticipate will happen in the first quarter of next year, and that's been a very important step. We also anticipate that the trial for multiple sulfatase deficiency will open early next year, or at least in the first quarter, but that of course depends on very critically on the ability to produce the drug, which is still ongoing. And that, as you know, can be a challenge with regard to AAV. But here again, along the lines of disseminating the information that we've gotten, we put together what we call the BGTC playbook that really describes the whole regulatory process of going through the idea for a first-in-human trial all the way up to dosing the first patient through the INTERACT and pre-IND and ultimately IND submission. The original version of this is fairly generic, and that's been made available and more recently made available in a way that rather than a static document, one that is available as a sort of click through. And what we plan to do is, as we develop some of these minimal packages and some of the learnings from that, from our interactions, make those available. There's some of those documents are already actually included in the BGTC playbook. So that's a public resource. And let me see. And then finally, I'll move to the somatic cell genome editing program that Shengdar mentioned. This is a program supported by the NIH Common Fund. The Common Fund programs support large cross-cutting efforts of interest to different parts of the NIH to do things and address challenges that no individual part of the NIH could do. And certainly the development and clinical development of genome editing falls into that category. Common Fund programs are funded for 5 years, and then there's an opportunity for a second 5 years. And we're now in the second phase of SCEG. The first phase that Shengdar was involved with and many others really focused on tool development. This started about 7 years ago. On tool development, new vectors, particularly new delivery systems, et cetera, to kind of lay the groundwork for moving into the clinic. But as we all know, things moved into the clinic very quickly. So in phase 2, we are trying to do the same thing. There's some efforts to develop new delivery systems, but really a lot of the focus is how do we move things through the regulatory process and ultimately into the clinic. We have several different areas that we're funding, including an initiative focused on basic, or not so much basic, but assays and the development of assays in a way that would make them fit for regulatory purpose. And Shengdar has been funded under that for the development of better and more robust ways to detect off-target editing. But we also have several of these more clinically focused programs, as you can see here. These are all types of awards that are called U19 awards, where in each case there's the focus is on a specific disease or a specific tissue target. And in each case, there's kind of a lead clinical asset as well as several other followers. And the idea is that within each of these programs, the groups can learn from each other. We've got several neurologic diseases, one more focused on metabolic disease and one focused on retinal disease. And one of the things we really hoped for in this program is that we put in requirements for each of these groups to have these meetings with the FDA, these INTERACT pre-IND meetings. And for these projects, we ultimately wanted them to get all the way to the point of an approved IND before the project was over. But one of the things we've really liked about this program is that the interactions between the different investigators. And I'll talk a little bit more about that later. This is just kind of one example of some really nice science that was done. It actually kind of overlaps work that began in SEG phase one. One of the projects from phase one was work done by Ben Deverman at the Broad Institute to try to develop a type of AAV that would efficiently cross the blood-brain barrier. Several publications, he identified the human transferrin receptor as a target that would allow crossing the blood-brain barrier. But he demonstrated that in a way I think was really very clever, as you can see on the left. What he basically did was to show that with his transferrin targeted vector, you get very little delivery across the blood-brain barrier into the brain in a normal mouse, but if you essentially humanize that mouse by making it overexpress the human transferrin receptor, you get much, much greater uptake and crossing of the blood-brain barrier, which again, I thought was a very sort of clever approach, this way of humanizing mice. And then as the group moved into phase two, Ben collaborated with Sonia Valab from the Broad Institute, who's very much focused on developing genome editing, epigenetic editing for prion disease. That's a autosomal dominant disease, and the idea is to use genome editing, epigenetic editing to silence the prion protein genes in a way that is described there with the acronym. And they had some really nice data using Ben's technology to show that they could, in fact, effectively epigenetically silence the mouse version of this gene, which is a really exciting way to treat not just the particular prion disease, but indeed, I think as a nice therapeutic platform for multiple autosomal dominant prion and other diseases. And just as I'll stop there, I do think that, as I said, we required all these groups to go through the INDs or the interact in pre-IND meetings. And what's been really nice is that each one of the groups have done that, they've worked together with others. So it's kind of a very interesting kind of collaborative approach to studying the regulatory path. It's whereas every interaction with the regulators can be built on, and for the next group going in. And a key part of that, of course, is coming up with a consistent approach for detecting off-target editing. And here it's been great to have Shengdar part of the group, who's made major contributions to all of those interact packages and other regulatory packages. So just to kind of tie all these things together, what you see on the top there was my very naive and oversimplified view of AAV gene therapy when we started with PAVE-GT and the BGTC. I kind of envisioned an AAV as sort of like an empty delivery box that was automatically pre-addressed to go to certain cells and tissues, and you just drop your therapeutic gene into it, and you get your vector, and you kind of move forward. It turns out that this is inaccurate for a variety of ways. In particular, it's the production of AAV vectors is not simply just dropping them into a box. All of the parts of the vector, including the outside parts of the box, are all made together in human cells in a way that can be very sort of challenging even today. But the other thing is that I think is kind of important as a distinction between typical AAV gene therapy for individual diseases versus gene editing is sort of the modularity of it and how you deal with multiple genes, because or multiple diseases, because in the traditional AAV gene therapy, you put one therapeutic gene into each time you make a vector, so you're always sort of doing one disease at a time. But gene editing offers a more modular approach that I think has very important implications. And so if you think about what you need for gene editing, you really need three components. You need a genome editor, which can be a messenger RNA encoding a genome editor. You need a delivery system, and you need a guide RNA, at least if you're doing Cas9-based gene editing. And this at least opens the potential that as we realize that once you have success with a given set of these components, to go for multiple, add multiple patients, multiple targets, and maybe even multiple diseases, you could just reuse the editor and the delivery system and only change the guide RNA. And that, if we could get to that point, seems like it'd be a big breakthrough in terms of efficiency and getting into first-in-human trials. So as part of the SCG phase 2, we actually funded several projects that actually required investigators to do that, to explore the regulatory pathway to develop gene editing as a therapeutic platform using one editor, one delivery system to treat more than one disease, and essentially learn by doing and interacting with the regulators about the potential of developing that platform approach. We funded one project that is scheduled or focused to go all the way into early clinical trials. This is a very interesting project from Yale University, focused on a non-viral CRISPR protein that is chemically modified and can be injected directly into the brain, where it goes into cells as a potential platform for treating neurogenetic diseases like Angelman syndrome and others. And we also have two other of these platform clinical trial programs. And one of those is focused on metabolic diseases, the one by Rebecca Ahrens-Nicholas and Kiran Musunuru, personalized prime editing to treat hepatic inborn errors of metabolism. Note that none of these are, even in the title, they don't mention individual diseases. They're all platforms. And for those of you who have been in the field, you've undoubtedly heard about one of the sort of honestly surprising and wonderful things that happened as part of the work that we funded Kiran and Becca to do. It's been about a year or so ago, actually earlier this year, kind of amazing. There was a baby born at Children's Hospital of Pennsylvania with a rare organic acidemia, a urea cycle disorder, I should say, the most severe form of that. And Karen and Becca were already sort of working on this heavily, and they were also participating in the SCG and having had now a lot of experience with the FDA and the regulatory path. So when this baby was born, the baby was very quickly identified as having this urea cycle disorder. They realized there was an opportunity to kind of move very quickly here and ultimately try to do a treatment. And through a wonderful collaboration that involved multiple groups, including industry partners and others, they were able to go from the molecular diagnosis of that baby all the way to the first in human dosing in about 7 months. And that included toxicology studies. We provide some extra funds for an animal toxicology study, but the time was really quite breathtaking, and very happy to say also that it seems from all indications that the baby who's baby KJ, arguably the most well-known baby in the United States, at least in my perspective, is doing quite well. I don't know exactly how you'd characterize his outcome, but certainly the treatment had very beneficial effects. It reduced the level of ammonia in the blood, which is a key thing you want to reduce in these urea cycle disorders. It's ultimately the ammonia in the blood that goes to the brain and causes brain damage. And they've also been able to take him off some of the dietary interventions. So a wonderful success, and one that was really a great boost to the field. And we were very, very happy about that. One thing, though, that I was a little had mixed feelings about, and this is of relevance to this particular group, was the sense that this was a great example of an N of 1. And that was not our goal in funding Kieran and Becca. the goal was actually to develop a therapeutic platform for genome editing. And very happily, they have moved on with their regulatory interactions. This is just kind of to say that there is this tension here between the common way we think of drug development as one disease at a time and these therapeutic platforms. There isn't really a good regulatory path, and indeed that's kind of why we funded the research to explore that. And so just a couple of months ago, they continued with their interactions about this platform approach for urea cycle disorders. They had an interact meeting with the FDA, and then in light of the success with baby KJ, they saw the opportunity to move very quickly and had a pre-IND meeting. And again, consistent with all of our efforts to try to take these regulatory learnings and put them in the public domain. They published this paper which describes their approach and the FDA's response to it about what they call the umbrella trial. And there's a lot of things to say about this, but the basic thing to me that was the most important is the FDA kind of agreed that this could be done with a single IND application for multiple diseases, and that the biodistribution toxicology study from one of the LNPs could be used to cover all of the different diseases. And that to me is really important because it means that you can add additional patients and even additional diseases without having to go through these very expensive, time-consuming animal toxicology studies. But you can read all about what was done in this paper. They have a link to another component of the SCEG where you can see their regulatory submission and the FDA's response and read it for yourself. But to me, very, very, very exciting work, and I'm looking forward to them moving ahead towards this trial as quickly as possible. So people might say, sure, that's great. It's easy to do with the liver. You know, you have everything goes to the liver. And we, of course, are aware of that. We want to extend this approach beyond the liver into other tissues. So another part of the SCEG program is still focused on better delivery systems, either ones that can be programmed or of particular relevance to neurologic diseases, non-viral approaches to cross the blood-brain barrier. So this is not a research grant, but a prize competition. And one of the areas of focus was, as I said, non-viral approaches across the blood-brain barrier. And we were able to identify 4 different projects where they had very compelling proof of concept studies to get a second prize in this multi-step prize competition. The next step for all of these will be to have, we're going to have these technologies independently validated in large animals. And based on that, we will identify a winner that will get, if they go all the way through, a total of about $1 million. And obviously very excited to see that. I guess the way we sort of think of it is the learnings about the regulatory path and the platform from Karen and Becca's work with the liver can be extended to other tissues once you've identified an effective delivery system. Now, of course, I know that many of you in this collaborative are focused on antisense oligonucleotides and not so much gene editing, and you're probably thinking, hey, what about antisense oligonucleotides? What can we do? And this is something that we think a lot about also. I think I personally and all of us, we're in favor of anything that works, that will be able to provide effective treatment for people with rare diseases. And one of the challenges that we're aware of is the ability, or currently the inability, to really accurately predict the toxicology of oligonucleotide therapeutics, including antisense oligos. And so thinking about that, one of the new projects we decided to kick off was what we call the Oligonucleotide Toxicity Open Data Challenge. It's going to be led by my colleague Eric Sid. And the goal of this is to try to identify data sets and ultimately incentivize the production of data sets that would be relevant to producing data to predict oligonucleotide toxicity in humans and make those publicly available with the idea of then sort of using them to feed data into AI-based approaches to come up with more effective ways to predict oligonucleotide toxicity. You know, we all hear about the great potential of AI. I'm certainly not an expert in AI, but I do understand that it's important to have high quality data to feed those. And that's really what we're sort of incentivizing. So there's the information about that, the key dates, et cetera. And obviously either I or Eric can happily answer any questions you may have. So just thinking a little bit ahead, this is a famous quote from Wayne Gretzky, the hockey player, where he said, "Some people skate to the puck. I skate to where the puck is going to be." It's a great quote. I have heard, though, there's some debate about whether it was actually Wayne Gretzky or his father who said it, but regardless of who it was, it's a great quote. And so he was thinking with regard to genetic therapies and treatments, where is the puck going? And I hope I've convinced you that we would like to think the puck is going towards therapeutic platforms rather than doing one disease at a time. And if that's the case, it does raise the question of how are we going to identify all of the patients with monogenic diseases who might benefit from these therapeutic platforms. And a few years ago now, actually during the pandemic, we had a series of discussions sort of anticipating this and thinking about it with the development of gene target therapies as they were then, and it involved discussions about newborn screening. And those papers got published. But more recently, we were able to, again, work with the NIH Common Fund and develop a new program called Newborn Screening by Whole Genome Sequencing, or NBS by WGS. And the goal here is really to assess the feasibility of incorporating a whole genome sequencing component into the current U.S. public health newborn screening program. And I'm kind of emphasizing that because that program, as many of you are aware, is currently focused on screening for, roughly 30 diseases. Involves taking a heel stick from the baby, getting those blood spots, and using typically mass spectrometry to study or screen for about, as I said, 30 diseases for which there are approved treatments. But thinking ahead, if we are going to be moving to these therapeutic platforms, we wanted to have a sort of a broader approach to that. As the NIH, we're not in the position of fundamentally changing the program, but what we couldn't do is fund a research study to assess whether it would even be feasible to incorporate whole genome sequencing into these current newborn health laboratory-based programs, which is going to be a big lift for some of them. These programs are very, very important. They provide a critical service. They are typically not so well funded. People work very, very hard. We don't want to damage these programs, but we do want to assess whether it be feasible to include whole genome sequencing. So that's the program we funded. We actually funded a group led by Robert Green and colleagues. You can see there, they're doing what's called the Beacon study. It just kicked off about a couple months ago, and we're very excited to see how that develops. And again, thinking about well into the future, where is that puck going, and see if we can help it connect up. So I want to mention that. And then finally, there's a question about how are we going to have and how are we going to develop a business model or business models for any or all of this. It's, again, a very different, think about therapeutic platforms as a very different business model. I'm not a business guy. I don't really have any good ideas about that at the current time. I did want to mention, I think, an interesting idea that is being developed now or launched in the United Kingdom, what's called the Rare Therapies Launchpad. And the idea here is that the UK regulatory authorities would potentially approve what they call centers of excellence, who would be able to do therapeutic platforms such as antisense oligonucleotides and potentially even genome editing. And once they got enough experience in doing this through various pilot projects, you might get to the point where there could be reimbursement, which I think is a really interesting idea. I have no idea whether that could work or how that could work in the United States or really about the business model at all. But I think as we will increasingly be thinking about these things in the future, I just kind of wanted to mention that as sort of food for thought. And again, I'm sure you're all aware of that. I've seen Tim on here, and he's been involved in this effort. And then finally, at the risk of causing too much offense, I think that even though this is the N of One collaborative, I personally try not to think of things and focus on N of one or ultra rare diseases or nano rare disease or any of these terms, in part because they're in some cases not so well defined, but more broadly because I feel like they're focusing more on the challenge and the problem as opposed to the solution. And I think what we're trying to support are solutions that involve the development of therapeutic platforms to treat monogenic disease, many diseases at a time. And that's kind of what I've been talking about. And so I'm happy to stop there and answer any questions. Thank you.
Great. Thank you, PJ, for a wonderful talk. I guess, let's see. I guess I see some hands raised. So why don't we kick off the questioning? I see Buma Yandava.
Thank you, Dr. Brooks. It's a wonderful presentation, bringing the entire field together, and you're truly one of the pioneers moving the regulatory landscape of rare diseases. Thank you so very much for this great presentation. My question is focused on, this is really great harmonizing the various regulatory pathway. I'm also wondering about the clinical pathway as well. Are there any initiatives that you think that can be focused on clinical pathway? Because one of the biggest the challenge is also identifying the patients and really bringing the patients together in terms of therapy and so forth. So do you have any thoughts that you can share? And the second question is also, the biggest challenge is commercialization. I mean, still the gene therapy is not something that it's affordable. So do you have any thoughts to share on making it more affordable for larger patient population? Thank you.
Sure. Well, thank you for the nice comments. I do want to try to be clear. I try to mention that all of these programs involve work by a lot of people at the NIH, and certainly not just me. I tried to put some of those names on the slides. In terms of the clinical approach, I think, again, sort of what Kieran and Becca are doing is going to be a nice example where you have people who work with groups of diseases that are sort of similar. In their case, these urea cycle disorders are more broadly inborn errors of metabolism. Again, I can imagine something like that with a group of neurodevelopmental disorders or neurodegenerative disorders. So I think that kind of approach will almost go in parallel. It almost has to go in parallel with some of the regulatory approaches. The commercialization one, I guess the only thing I can say is scientifically, therapeutic platforms make a great deal of sense. From the commercialization perspective, for reasons I don't fully understand, they don't. I think people and drug companies, I think this is just the way they think of it. They think of developing one drug to treat one disease, and they can calculate that return on investment. And I had that, slide up there, the square peg in the round hole. I feel like there's got to be some way to change the thinking, and I don't have an answer to it, but I think that I don't know. I'm either an optimist or naive or whatever, or maybe I'll be very disappointed. But I think that if the science makes sense, we will find a way to have uh, a business model. But like I said, it's not my job at NIH to develop those. But obviously, it's a really important idea. Thanks for the
Thank you
question.
so very much. Yeah, I can the platform, how important it is, it resonates with me as well. And my PhD thesis is focused on demonstrating the value creation abilities of platform technologies. And I like to follow,
Oh,
learn
oh,
from you, follow my conversation with you further
well,
offline.
I would like to learn from you, so please
Oh,
follow up with me afterward, because I'm all ears on that. Thank you.
thank you.
Tim?
Hey.
Hi, PJ. Thank you so much for that wonderful presentation and for joining us. I have a comment, which is and a small question. First, I just want to comment that um, how lucky I feel. Like I think that I remember our early conversations back in 2017, 2018, thinking about what was possible with the idea of using platform therapies and what NIH could catalyze. And I just wanted to say like how impressive it is to see what you, and I know you're going to deflect away from yourself, but I'll focus on you for a second, and also the royal you at NIH and NCATS have been able to set up in terms of these programs that have enabled folks like Shandar and Kieran and Eric and Sonia to advance these amazing scientific programs that are kind of building on that idea. It's really cool to see. So my question actually is, uh, with respect to thinking about what that next initiative might be that you launch with Beacon, with the PaveGT, with the SCGE and all of these. Are there opportunities for folks in this community to engage with you on discussing the trajectory of the puck and maybe offering ideas or learning from you about where you think the puck is going, or maybe offering some of our own thoughts into that, planning the next program that you're going to unveil?
Well, yes, I think that there will be, and I do appreciate the nice comments. I really want to give a shout out to the NIH Common Fund, who's been very supportive, and again, all the different NIH institutes that we work with, particularly NINDS and Tim Levote, who's my partner in crime with SCG. I think that we have thought about, and indeed we're working on having some meetings to get some of these ideas, because I do think that some of what we've seen so recently changed things quite a bit. And there have been other meetings, but I anticipate that we will be having meetings like that. I can't predict in terms of future funding or opportunities, as everybody is obviously aware. There's a lot of questions about NIH funding, and so I can't really say anything about what future initiatives might be coming down the line. But I think certainly hearing feedback from all of you who are kind of working in the trenches will be very valuable, and we will definitely be planning ways to do that moving forward, possibly even at ASGCT or maybe even a larger meeting after that. That's something we've been thinking a lot about. So yes.
If I might follow up then, like with maybe one of those ideas, do you think that you've got now platform therapies covered, you've got platform early diagnostics being covered by Beacon? One of the things I've wondered about, do you see a value in the platform development of biomarkers with which we can actually track target engagement across a multitude of different diseases that, with a similar or common methods? If we could turn, for instance, in my space, the brain into the liver, and to be able to pull out metabolic readouts of the diseases that we are working hard to try to treat. Neurofilament's great, but we could do so much more.
It's a really neat idea. It's literally something I've never thought about. but I'll certainly go back and talk about that with my colleagues and see. It certainly makes a lot of sense. Yeah.
Great.
Thanks.
I have a question. So, PJ, I wanted to just echo some of the comments. Thanking you for your leadership in this space. It's really great to see, I guess, all the different parts together, including like PAVE-GT, the Bespoke Gene Therapy Consortium, NIH SEG, and like this theme towards platformization. Regarding the idea of like, where is the puck going to be? I was wondering, like, where do you see the path to scalability related to gene editing? Is it making more of these like N-of-1 approaches more straightforward, so more like mutation-specific correction? Do you see like more mutation-agnostic approaches that could apply to more diseases? And I guess what do you see as pros and cons in those approaches?
Yeah, I think that as clearly scaling up will be an issue, but I guess what I'm thinking is as we see certain approaches become successful, we will see more people doing them, and you will then start to see sort of economies of scale. That would be my hope, and with regard to both the delivery systems and also I'll say oligonucleotides, because of course guide RNAs are oligonucleotides, as are antisense oligonucleotides. And I'm not so familiar here with the chemistry, but it sort of seems intuitive that as more people kind of focus on that and there's more progress, that we would see efforts moving forward to make those more affordable and scalable. That would be my hope. I'm kind of thinking way back in the early days of genome sequencers, right? That were machines. When people started doing that, all of a sudden you'd get people, 50, 100 machines in places. And I guess that's one thought. I hadn't really thought about that before you asked the question, but I think we're going to kind of see that some basic approaches turn out to be the best for certain cells and tissues. It seems right now that lipid nanoparticles are really good for getting to the liver. They may be good for other tissues as well. As you're aware, some really impressive work that also came out of CHOP, done by Bill Parento. Some of these lipid nanoparticles are really very effective at delivering to muscle if the delivery is done prenatally. And it may be that as we see more approaches like that, the key difference that I've learned between AAV and the lipid nanoparticles is one that, Peter Marks had kind of explained to me, is like lipid nanoparticles, messenger RNAs, these are all really chemical synthesis. Making an AAV is making a biologic, and it's a pretty complicated biologic. So I think there's a lot of benefits in trying to develop an approach that is, synthetic chemistry. But will that work for everything? I don't know. I think the blood-brain barrier is a huge challenge. Some of the work that's coming out is very exciting. I mean, the stuff that Ben Deverman did. So I think we'll see. But ultimately, I feel like the field will kind of coalesce around a limited number of technologies, and then maybe we can really benefit from economies of scale.
Great, thanks. Let's see, other questions? Otherwise, I have a lot more as well. Are there some in the... Okay, so I had a question related to, I guess, like the Bespoke Gene Therapy Consortium, and how are you able to kind of get this like public-private partnership to be open to kind of releasing kind of regulatory information, or to develop this playbook? So I guess, how was that idea formed, and like, how could it be replicated?
Yeah, I mean, the whole idea of doing these, the concept is really of the public-private partnership, is that's what the Foundation for the NIH does. I think the interesting thing about the BGTC is we decided early on that we had to work on diseases that were all of which are so rare that they're of no commercial interest. And so that automatically sort of produces what you might think of as a pre-competitive space. And in that context, it's really kind of fun. We got people who have expertise in different aspects of the regulatory process, just kind of sitting around thinking about what would make the most sense. And because there's no proprietary information, we can put it out there. I think that might be a challenge to replicate. Early on in the BGTC, we thought about maybe we could do something around better AAV serotypes, but we realized no, because all of these companies are working on that. That's proprietary. Understandably, they're not going to just share all their cool data. So I think that it's part of the way it was designed to focus on those diseases of no commercial interest that allowed everybody to kind of work together. And it really is kind of, in that regard, a great sort of learning collaboration. And people really, it's amazing how much time people spend on this, given that they all have, full-time jobs, right? This is all the people in the BGTC are volunteers, for the most part, the people from the company. So I think that has a lot to do with it.
Yeah, I see. Okay, that's super interesting. I guess related to kind of like that commercial interest and no commercial interest idea, I think that in the gene editing space, it seems like there's been a lot of focus. Like a lot of people are working on the same thing, right? There's like a lot of people working on, for example, hemoglobinopathies and things like that. And so there's kind of like this clustering. And obviously, I think like, personally, there's in this whole group, like the N1C, very interested in rare disease therapy. But as you mentioned, like, not as much commercial viability. And so I guess like how do you get a balance of things? How do you incentivize people to work on more of these rare disease or like these platform ideas?
I don't know. I mean, that's what all I can say is it just seems to make such obvious scientific sense. And I keep thinking that eventually there'll be a way to develop a business model for what seems like obvious scientific approaches. But I think that's one where, kind of in response to Tim, I think we want to keep having some conversation about this, because I don't have that answer. I'm just naive. But I think that's ultimately the biggest question. So, interested to hear. And also, one thing I will say is I think we've seen very recently now the idea of sort of non-traditional entities marketing drugs. Just last week, we had this great announcement from the San Rafael Telethon, getting approval. I think entities like that have a different sort of business model coming together. I don't have the answer, but that things like that sort of I find very encouraging, and I think that the program, the Launchpad in the UK, I think is really interesting. But would that work here? I don't know, but yeah, I'm afraid the best answer I have.
Great. Is there anything we haven't covered that you want to add, BJ, about your overall vision for the future?
No, I think I've said more than enough about my thoughts on this. So yeah, I think I've said everything I need to say.
Okay, terrific.
Maybe
All right.
I'll add one thing, which is, if I may, Shankar, sorry,
Yes, please. Yeah.
to PJ's last comment about the uses and the interpretations of N of one, I completely actually agree with you that N of one was sort of where maybe this group all started, and I think we all aspired to this being really about platform medicine. I think that was the vision. And N of one may have been the term that was useful at the time to encapsulate the problem, but it's not the solution. The solution is the programmable platform approach. So you will see us saying exactly that same thing. If there are discussions about rebranding at some point, we'll ask you what you think. But just to point out that we're completely aligned with you on that.
Yeah, I think everybody is. I think it's just a term that we're all kind of comfortable with, and it literally started with an N of one. To me, it started with Mila, right? So, but yeah, I think that's the way to go, and I think everybody working in this field kind of understands that.
Great. Okay. Are there any other final questions or remarks? Okay, with that, I'd like to thank the N1C organizers for organizing a terrific thing, PJ for giving an outstanding talk and discussion. Thank you all for attending. We're invited to continue discussion on N1C connections, and please complete the survey, which will pop up as we end. Thanks very much.
Thank you.
Thank you.
Bye, everyone.